Okay, so I'll go ahead. Thanks to Eric for inviting me to speak today. So I'm Ben Spurgeon, and I'm a postdoc at the Center for Platelet Research Studies at Boston Children's Hospital. And, we use a variety of different techniques to examine platelets, and we're currently using mass cytometry to explore platelet heterogeneity in health and disease. And in today's presentation, I'm going to talk you through some of the work we've been doing on this. So I'm guessing that some of us today are not too familiar with platelets, so I've prepared a quick overview slide. Platelets are the smallest blood cell at around 1 to 3 micrometers, but they're very highly abundant with a normal range of 150 to 450,000 platelets per microliter of blood. And a healthy adult typically has over 1 trillion platelets in the circulation. And platelets have a lifespan of around 10 days, and then they're removed from the circulation, predominantly by the spleen, but also by the liver. And they're continually released into the bloodstream by megakaryocytes, which reside mainly in the bone marrow, but are also present in the lungs. And megakaryocytes are themselves very heterogeneous, and we think that distinct megakaryocyte subpopulations release distinct platelet subpopulations into the blood, and so platelets can be born different, but platelets can become even more different throughout their lifespan because platelet phenotypes can be dynamically modulated by many different substances that are present in the bloodstream, from cytokines to lipids, bacteria and other pathogens. And the primary role of platelets is to prevent bleeding, and platelets can become activated very, very rapidly at the site of hemorrhage, where they aggregate with one another and contribute to formation of a hemostatic plug or a blood clot. But in recent years, a number of alternate roles have been identified for platelets, including roles in immunity and inflammation. So platelets express a wide range of receptors that allow them to perform specific functions. And we designed a panel of antibodies that would allow us to perform a comprehensive evaluation of platelet phenotypes and functions. And this panel, it's a 14 marker panel shown in this table. Many of these antibodies are commercially available. Some were obtained from the local CyTOF core, while others, such as GP6 and PAC1, they were labeled in-house using the MaxPar labeling kits. And it is a relatively small panel of 14 markers, but spillover exists in several channels. And the MaxPar panel wheel shown here in panel A, it did a good job of predicting this spill and suggested that some of our markers, including CD107A, CD62P, and CD63, might be affected by spill and might have a low tolerance for it. And we confirmed this by running compensation beads through the Catalyst app and generating a spill matrix, which showed that CD107A, CD62P and CD63 were indeed affected by spill. It was small, but CD62P received the most at 3.8%, which is potentially problematic because CD62P is a very important marker of platelet activation. And if this spill was left uncorrected, it might lead us to believe that platelets were activated when in fact they are not activated. And so the heat map in panel C shows the pre and post compensation signals for for all of the markers. You know, the spill is small, so some of these corrections are barely visible, but we can see that Catalyst did a good job on CD62P, where the compensated signal in the basal sample is extremely low, which is exactly what we would expect and makes you know, perfect biological sense. You know, so the takeaway is that, you know, our panel's small, but it contains some potentially problematic spill. This spill can be corrected by compensation. And we routinely run all of our files through Catalyst prior to analysis to perform compensation, although I'm now aware of, I think there's an alternate compensation tool called CytoSpill, but we've not tried this, this new approach. So the following slide gives an overview of the basic method. You know, we collect blood from, you know, typically healthy donors. Sometimes we deal with patient samples. And after we draw this blood, we pipette 3 microliters into an antibody cocktail that may or may not contain agonists which activate platelets. You know, we stain for 30 minutes at room temperature and then add a lyse fix buffer containing iridium cell ID. We then wash the samples twice in milliQ, resuspend in EQ beads and filter for acquisition by mass cytometry. So once the FCS files are received, we perform the compensation and then we gate the single platelets according to the following steps. So in step A, we separate the cells from the EQ beads. We then gate platelets based on CD61 expression. So the platelets appear as iridium low and CD61 high. And finally, we gate the singlets based on event length. And then we export these gated singlet files for further analysis. So now we'll get into the experimental data that's shown in the paper. And the first figure shows the effect of anticoagulant choice on the expression of our 14 platelet markers. So the anticoagulants sodium citrate, heparin, and EDTA are very commonly used in platelet studies. And there's often, you know, some debate about, you know, the optimal choice. It's worth noting that calcium is essential for platelet function and coagulation, and citrate and EDTA work by chelating calcium, whereas heparin works by inhibiting the activity of thrombin, which is also essential for coagulation. So to determine the best anticoagulant, we collected blood into citrate, heparin and EDTA tubes and added this blood to antibody cocktails with or without platelet agonists. And we used the combination of ADP and TRAP as platelet agonists in order to induce the expression of all 14 markers, because some markers are very, very low or absent on the surface of basal platelets. And generally, we found that staining was comparable for most markers across the 3 anticoagulants. But down, I think in the last plot for PAC1, EDTA completely inhibited the PAC1 signal. And PAC1 is a monoclonal antibody that recognizes the activated form of integrin alpha-2b beta-3. And this integrin is essential for binding fibrinogen and allowing platelet aggregation. And calcium is essential for integrin structure. So it's likely that EDTA is chelating calcium at a level that disrupts the structure of this integrin, so that PAC1 is unable to recognize the epitope. So we determined that EDTA was incompatible with the current panel. Heparin does allow reasonable staining, but some markers, including PAC1, were slightly, although not significantly, elevated in basal samples collected in heparin. And this is consistent with previous reports that heparin might cause spontaneous platelet activation. And so we decided to perform all subsequent assays using sodium citrate as the optimal anticoagulant. So the next figure in the paper, we look at antibody cocktail storage, and for convenience, it's often desirable to use pre-made antibody cocktails. And so we wanted to assess the effect of cocktail storage on the staining of our 14 platelet markers. So we made up some antibody cocktails and stored them at 4 degrees or -20 degrees for either 2 or 4 weeks, and then compared them to freshly made control cocktails. And staining was consistent for all for the majority of markers, except CD61, which was significantly lower across all storage conditions. But these reductions in CD61 staining did not affect our ability to identify or gate bona fide platelets. CD63 and CD36 were also somewhat lower in some samples, while CD40B2B was significantly higher after cold storage for 4 weeks. And staining variation after prolonged cocktail storage, I think it's been reported in several publications, and some solutions have been proposed, such as storage at -18 FBS and DMSO, as well as preparation of lyophilized cocktails, but we've not yet tried these suggested solutions. And so figure 3 shows the effect of sample storage on signal intensity. So it's not always possible to analyze samples immediately, especially when samples are prepared off-site. So the storage of stained and fixed samples is sometimes necessary. And so we wanted to assess the effects of sample storage on the staining of our 14 platelet markers. So we prepared basal and stimulated samples from 3 different blood donors, split them into aliquots for either same day analysis, which is the control, or for analysis after storage for 2 and 4 weeks at 4 degrees or -20 degrees. And we found that the cold storage of stained and fixed samples was associated with significant variation for multiple markers. Some of these markers, including CD41 and CD61, were significantly lower after storage, while others, including CD107A, CD63, and PAC1, were significantly higher. And we were quite surprised by this, but I think some publications have suggested that prolonged sample storage might result in the exchange of metals between antibodies, and this could potentially explain the increase in signal intensity that we're seeing for some markers. So in figures 2 and 3, we've identified, some sources of variation. I think the paper could have been stronger if we were able to demonstrate an approach to correct for this variation, but unfortunately, we were unable to do that at the time. We've since made some progress in this area, and there's a couple of slides that I might show at the end of the presentation, where we've attempted to address this issue, and that might spark some discussion. And so figure 4 in the paper, we did an experiment looking at cell transmission. So compared to conventional flow cytometry, CyTOF is known to have a lower cell transmission efficiency, which is potentially problematic for our laboratory because we sometimes receive samples from patients with a disease called thrombocytopenia. And patients with thrombocytopenia have very, very low platelet counts. And these platelet counts can be as low as 2,000 platelets per microliter versus, 300,000 platelets per microliter in, a normal adult donor. And so in these particular samples, it's important that we were able to analyze as many platelets as possible. So we wanted to optimize our protocol to maximize the transmission and detection of single platelets. And so we mapped platelet transmission throughout the entire protocol, first by looking at platelet loss after cold storage, and then at platelet loss after washing. And in these experiments, the platelet counts were performed by flow cytometry. So if we look at figure panel A, we can see that stained and fixed samples stored for 4 degrees overnight did not differ from the controls, but samples stored at -20 degrees overnight resulted in a significant 11% loss of platelets, which might be due to freeze-thaw lysis. And significant cell loss was also seen after washing. So if we look at the bar graph next to this, W1 is the first wash, W2 is the second wash, and the control is the platelet count prior to washing. So we saw a significant loss of 20% after 1 wash, while 2 washes resulted in a cumulative loss of 27% of platelets. So we're losing, at least 1/4 of platelets prior to acquisition on the CyTOF. And to determine platelet loss during acquisition itself, we stained platelets with a fluorescent platelet identifier for flow cytometry counting, plus iridium and CD61, which are used to identify platelets by CyTOF. We then prepared 2-fold serial dilutions of samples from a maximum of 4 million platelets per milliliter all the way down to 31,000 platelets per milliliter. And we acquired these serially diluted samples by CyTOF. And the FCS files were exported with different settings for event length or minimum event duration and lower convolution threshold. And we calculated the cell transmission efficiency across all of these different samples and settings using the formula that's displayed on the screen. And because we know the concentration of cells in the sample, and because we can acquire a specific volume of that sample, I think the flow rate is known to be 30 microliters per minute. We can therefore calculate the number of platelets that were introduced into the CyTOF instrument. And we can divide this number by the number of single platelets detected, and multiplying this by 100 gives us the transmission efficiency. And we plotted these results in figure panel B, which shows that transmission was generally highest in samples that were concentrated at 250,000 platelets per milliliter. Although, transmission was fairly good all the way up to about 1 million platelets per milliliter. Transmission was also highest with a lower event length limit of 5. And the literature suggests that event length is dependent on iridium staining, and because platelets are very small anucleate cells, they bind less iridium and therefore generate very low event lengths. So the default event length limit on the CyTOF, I think, is 10, but this appears to exclude some platelets and therefore allows detection of fewer singlets and results in lower transmission. And we can see this in panel C, which is just a visual representation of the data from lowest transmission to highest and something in between. And we can see that the lower event length limit of 10 seems to, there's kind of a hard edge here. It seems to, cut off or obstruct the detection of some platelets. And if we look when we change the lower event length limit to 5, we get an additional detection of platelets here that's not chopped off. And so we found that the maximum transmission efficiency was 68% under the optimal conditions. And when taken together with the protocol-related cell loss, we expect to lose about 50% of platelets from the initial sample. And this knowledge is really useful to us, especially when working with patients who have low platelet counts, because we can, kind of scale up and adjust as necessary in order to make sure that we're requiring a sufficient number of platelets. And so, this was the final figure in the paper, and, we wanted to wrap up just by demonstrating the application of clustering and dimension reduction to our platelet samples, and to, kind of illustrate the heterogeneity across different sample types. And we used omic to run FlowSOM using the elbow consensus to identify the optimal number of metaclusters, and we overlaid these metaclusters onto a t-SNE plot. We identified 4 metaclusters, which isn't many, but we're dealing with, a limited number of donors. This is from 3 donors, all healthy, and a limited number of sample types, basically, basal, ADP, and TRAP. So therefore, we might not expect to see much heterogeneity, but nonetheless, metacluster 1 was low for the expression of platelet activation markers, which were CD107A, CD62P, CD63, and PAC1, and these clustered together on the heat map. And not surprisingly, this metacluster was highest in the basal samples, where you would expect platelet activation to be low. By contrast, metacluster 3 was high for the expression of activation markers, and this metacluster was highest in samples treated with the strong agonist TRAP, which makes sense. So, I think this is a, kind of just a basic application of these tools, and we've since put a lot more work into optimizing clustering and dimension reduction for platelet analysis, and we've seen some much better results since this was published. So to summarize, we've shown that citrate anticoagulation allows optimal staining of our platelet markers, and that antibody cocktails and stained and fixed samples show some variation after cold storage. We've also shown that platelet loss occurs after the freezing of stained and fixed samples, and also after washing. And we've shown that sample concentration and instrument settings can affect the detection of single platelets. On the previous slide, we also demonstrated the application of multidimensional tools for the analysis of platelet samples. And now I'll just skip to a couple of slides in the appendix to show some, some recent things that we've been working on. So I mentioned that, some of our samples were, affected by, variation, and I think CyTOF is known to be susceptible to batch effects. One of the tools that we've been looking at to correct for this variation is called Fda Norm. And this is available on Omic, which is where we perform this analysis. And so if, just looking at these graphs, we've got 3 different batches of samples. Batch 1 is blue, 2 is orange, 3 is green. And this is just for one of our, probably the worst affected marker, which was CD42A, which was, very, very high in batch 1 and much lower in batches 2 and 3. And after we put the data through Fda Norm, this kind of, normalized the signal across all 3 batches. And, we're still kind of evaluating this tool. Maybe you guys have, know about other tools and other approaches that might be effective, but it seems to give us, reasonable results. We run a t-SNE plot on the different batches, and not surprisingly, each batch mapped to a different region of 2-dimensional space when the raw non-normalized data was used. But when after normalization, the results were much more consistent across each of the different batches. So it seems like, this could be a possible solution to address the batch variation that we're seeing in some of our samples. And that, I believe, is the end of the presentation. So thank you.
Thank you, Ben. Great presentation. We'll open it up for questions for Ben.
Hi, Ben. I had a quick question. So when you have the variation in your sample prep, it's post staining or pre-staining or both? Because I'm wondering if you could use a little bit of DMSO, because normally for like freeze thaw, we store cells with a little DMSO to help with that transition. And you had mentioned that, and I hadn't heard about this before, potentially storing some of the antibody cocktails with some DMSO to keep it stable. So I'm wondering if maybe you could keep a little DMSO in.
Yeah, I think that's potentially worth looking at. There was no DMSO in our samples. We just added the lyse fix buffer to the stained samples, and then we put those fix samples straight into storage at either 4 degrees or -20. So there was no DMSO added to those, but it might be worth giving it a try and seeing if we can improve things.
Cool, thank you.
Thanks.
We have a question from Greg for you, Ben. Do the marker expression profiles seen via CyTOF agree with other proteomic assays that may have been performed on these samples, either concurrently or historically?
Yeah, I think the initial paper that our lab published on this before I came to Boston. I think they did a head-to-head comparison with flow cytometry, the fluorescence flow cytometry measurement of the same markers, and there was a good agreement between CyTOF and flow cytometry, for the measurement of these markers. And even so, I think everything, all of the results were seen by CyTOF. They make perfect biological sense. The markers that we would expect to be low at basal are low at basal, and the ones that we would expect to increase with specific agonists, they do increase with specific agonists. So, I think, the results do make sense. And, as I said, it's been cross-validated against, established flow cytometry techniques.
Go ahead, Zhao.
Oh, hi. Thanks, Eric. Hi, Ben. Thank you for your speech. And I have 2 questions for you. So the first one is, the platelet is kind of like is small size, and it has kind of like, from the size point of view, it has a lot of overlap with other like subcellular organelles like extracellular vesicles, so those kind of small vesicles. How do you, when you define the size of the platelet, like when you're setting the parameters on the instrument, how do you fine-tune those parameters? And the second question is, I noticed that the antibody cocktails after you make the antibody cocktails and then frozen them, is kind of like increases. Some markers are increased expression levels. Do you know the possible reasons for that? Thank you.
Okay. So for the first question, platelets are, they are very small, and but they are still, much larger than, microparticles. And in our experience in our lab, we find that, the washing steps tend to remove most of the particles that are smaller than platelets. So, including microparticles, but nonetheless, we using the platelet marker, the platelet specific marker CD61, which is highly expressed on the platelet surface, that kind of allows us to resolve or separate the platelet population from any other particles present in the sample that might not be platelets. So, that's kind of, how we deal with that issue. And, all of the CD61 positive events, they look like, bona fide platelets. We've got, 13 other markers to examine for this population, all of which are expressed on the platelet surface, and all of which behave as we would expect them to behave on the surface of the platelets. So, we are quite confident that, the population that we're gating, we are very confident that those are bona fide platelets and do not include any other, particles, microparticles or microvesicles. And so to the second question about the variation in signal intensity and the increase in signal that we see after storage of our antibody cocktails, just looking at some of the publications in the literature, I think some groups have suggested that there can be an exchange of metals between antibodies when cocktails are stored for a long time. So, some metals could fall off of, one antibody and attach themselves to, another one. And if that other marker is much more highly expressed, then you might expect to see an increase in signal. So, I think that's potentially, one thing that's happening in there, although, maybe, some other things are going on as well that we don't know about.
Okay, thank you.
Thanks.
Any other questions while we're here?
I have a comment. So I know that if you stain your samples and you then store them, one thing that can happen that may increase your signal is because proteins are flexible, and like when we think about molecular modeling of proteins, oftentimes you forget about all the water that's in between. When you freeze, the change in orientation that happens just because of the condensation can allow some epitopes to be more available. It also works in the reverse way. Sometimes if you freeze, those epitopes aren't as well exposed and you get a decrease in signaling. So that can contribute as well.
Okay, yeah, no, that's not something that we've considered. So that's a great comment and something that we can consider. Thanks.
Sure.
You have another message from the chat, Ben, a question regarding the platelet activation. How can this be controlled during the procedure of sample preparation?
Okay, yeah, that's a good question. And, I think, once platelets are taken out of the body, they are susceptible to, spontaneous activation. The procedures for, drawing platelets have been, well standardized for a number of decades now. In order to minimize, platelet activation that results from, venipuncture or, storage. So typically, in order to, prevent platelet activation, we use a wide bore needle for the blood draw, which kind of reduces the pressure of the flowing blood, and we also discard the first few milliliters of blood, which may be which may contain platelets that were activated from the initial venipuncture wound. And then, obviously we're using anticoagulants, which kind of keep platelets calm under the right conditions. And also we strive to stain the samples within 30 minutes of the blood draw, because the longer you leave the blood sample standing, the more activated the platelets become, but also the longer you leave the samples standing around, the less responsive platelets become to agonists. And so our group and many others have determined that samples should generally be initiated within 30 minutes, especially when you're looking at platelet activation markers. So we kind of follow, that, these well-established techniques and procedures for, initial blood draw to making sure we stain the platelets within a reasonable time.
Thank you. I had a question personally. In your freezing of the cocktail and your fridge preparation of the cocktails, were those antibody cocktails just mixed in like a CSB or PBS solution, or were they in antibody stabilizer?
Yeah, they were in EP saline with BSA and I think 0.05% sodium azide.
Okay.
Dr. Chen from Mayo, he had to leave, but he did have a question in the chat about how fast the assay is.
Ben, did you catch that question?
The assay time.
Let me. I need to open the chat. So. Trying to find the question.
He was just curious on how fast was the assay.
Oh, okay, yeah.
The assay, yeah.
I found it.
Yeah.
So, 30 minutes, staining. Our spin steps. After washing, we do 3 spins of 30 minutes. Which takes us to 2 hours. And then we're kind of ready to take those samples across to the CyTOF core for analysis. Obviously there's some pipetting steps that take place in between the spins. So we're generally looking at 2.5 hours from blood draw to the time that the samples are ready for analysis. So it's fairly quick.
Ben, have you guys experimented with doing any sort of fixation to try and account for some of the sample storage concerns that you saw?
Trying different fixatives and fixing conditions, do you mean?
Yes.
Yeah. No, we've just used the standard dilution, the manufacturer's recommended dilution of the lyse fix buffer, and that's what we've stored the samples in. We've not experimented with increased concentrations of formaldehyde or lower concentrations. I think the final concentration of formaldehyde is 4% in the samples, so the samples are stored in 4% formaldehyde, which is, in our experience, I think it's more than sufficient to fix platelets. Platelets, we typically fix with 1%, and that's good enough.
Okay, yeah, that makes sense.
I'm curious about, I just have some expertise in platelets, not really in cytometry. A few of us here from the special coag lab at Mayo. As far as platelets go, pH can be sort of a concern when you're pre-analytically handling them. I was just curious if some of the stains, within cytometry process are looked at for that, as the effect on cells over time with open tubes, or you said a few 30-minute spins. If there's a concern that maybe the pH could change on these things and that would maybe be a variable construct to the sample performance?
It's possible. So, prior to fixation, we're keeping the platelets, at a physiological pH. The staining buffer that we use is a physiological pH. But once the cells are fixed, obviously, they're then taken through wash steps. But I think the pH of the molecule water, that's near physiological, and I'm not sure if that would disrupt any of the staining. I'm not sure what the effects of that might be. Again, it might be worth looking at that, but I'm not sure what the solution would be, because I think although you can wash in the you can wash the samples. Eric probably knows the answer to this. Can you do the washes in the new MaxPar buffer?
Yeah, and the CAS, that might help. It has a little bit more physiological pH.
Yeah, so I mean, that's a good question. So maybe doing the washes in molecule water is kind of having an effect, and maybe we can compare this with the the MaxPar washing buffer to see if there's any difference there. And that's not something that I'd previously considered. So thanks for that comment.
I can just from my experience, I think the cell acquisition buffer, the CAS solution, is a little bit acidic side compared to water, but still both water and you use water or CAS solution, it doesn't make a big difference if you stain your sample after fixation. After fixation, whether you use water or use CAS solution to wash or during the acquisition, it doesn't make that much differences. But I prefer to use CAS solution. Sometimes if you have some debris or particles stuck in the tubing system, instrument tubing system, it's just it's better, a little bit better compared to water. So, yeah.
Thank you.
I think we're going to have to make that switch because I think the CyTOF core in Boston, they've just upgraded to the new instruments, and that requires the use of this new cell acquisition buffer, so we can no longer acquire our samples in water. So I think this is an experiment that we're going to have to do at some point soon.
Yeah, and using the Cas9 buffer mitigates any potential issues of barium, which does appear in milky water, unfortunately. I had a question about there was a comment that the platelets are pH sensitive. So if that isn't just a purely chemical response, like a mechanical response, once they're fixed, it shouldn't work, right?
Yeah, I think once the platelets are fixed, I'm not sure what the effects would be on the staining. Prior to fixation, I think pH does affect platelet function. So if there are any alterations in pH, platelets might not respond to specific agonists, or they might undergo spontaneous activation. And I think one of the if for many groups who perform platelet isolation, so if they want to isolate washed platelets from whole blood, one of the techniques they use is to reduce the pH down to about 6.5, which is a technique to prevent platelet activation during the cell isolation procedure.
We have another question in the chat from Greg. Are there any proteins expressed intracellularly on the platelets, and are they of interest to interrogate, or is intracellular staining even possible with platelets?
Yeah, so, you know, kind of, I did my PhD using, you know, flow cytometry and fluorescent cell barcoding to stain intracellular markers in platelets. And it, you know, it is possible. You know, it's kind of difficult to get strong signals from platelets for many of the intracellular markers. So panel design is, you know, quite can be quite tricky. But you know, there is an intracellular marker called VASP, or vasodilator stimulated phosphoprotein, that's quite highly abundant inside the platelets. And there is actually a commercially available diagnostic test for flow cytometry, which aims to measure VASP phosphorylation because it can be used to assess how effective some antiplatelet drugs are. So, you know, the level of phosphorylation of VASP inside the platelet can give us clues as to, you know, how effective a particular antiplatelet drug might be. So yes, you know, intracellular platelet proteins, you know, they do have, you know, clinical significance, and I think there is some value to measuring these, although we've only done this so far by flow cytometry, and we've not tried it by CyTOF, although, you know, that's potentially, you know, something we could do in the future.